In this section, we will discuss about mechanism of transplant rejection and role of HLA molecules in this process. We can classify transplant rejection based on mechanism of rejection and time since transplant. Mechanism of rejection can be cellular rejection. In this type of rejection, recipient cytotoxic cells result in damage to transplanted organ. Or it can be humoral or antibody mediated rejection. In this type of rejection, antibodies against donor HLA molecules and other proteins result in damage to transplanted organ. In some cases, it can be mixed type in which both cellular and humoral components are present. We can also classify transplant rejection based on when it occurs after transplant. Hyperacute rejection occurs immediately after transplant and may become apparent during transplant procedure. Acute rejection commonly occurs few days to few months after transplant, while chronic rejection occurs few months after transplant and can occur anytime after this. In diagnosis of transplant rejection, we actually combine both these parameters. So acute rejection can be either acute cellular rejection or acute antibody mediated rejection. Similarly, chronic rejection can also be either chronic cellular rejection or chronic humoral rejection, while hyperacute rejection is only antibody mediated rejection. Transplant rejection actually results from immune response against donor HLA molecules and other proteins present in transplanted organ that are different than recipient. This immune response is comparable to immune response for infections. In case of infections, first there is activation of innate immune system. Then antigen presenting cells present antigens derived from pathogens to T cells. This results in activation and proliferation of T cells. Then effector mechanisms include either cell-mediated or cytotoxic response that helps in eradication of viral infections or infections with other intracellular pathogens. Or it can be antibody-mediated response that helps in eradication of infections with extracellular pathogens. Immune response that results in transplant rejection is also similar. First, there is activation of innate immune system. Then recipient T cells recognize donor HLA molecules. This process is called allorecognition. This results in activation and proliferation of recipient T cells. Effector mechanisms include cell-mediated response that results in cellular rejection, or antibody-mediated response that results in antibody-mediated rejection. Now let's discuss different steps involved in transplant rejection in some detail. In first step, there is activation of innate immune system. Every organ at time of transplant suffers degree of ischemia and then reperfusion injury. This reperfusion injury results in production of oxygen-free radicals. These oxygen-free radicals result in damage to parenchymal cells of transplanted organ. These cells then release molecules called damage-associated molecular patterns like heat shock protein. This process results in activation of complement and inflammatory response. These damage-associated molecular patterns are recognized by pattern recognition receptors present on dendritic cells. This results in activation of dendritic cells. Dendritic cells then leave transplanted organ and migrate to regional lymph nodes, where these cells will interact with recipient CD4 and CD8 T cells. These dendritic cells have donor HLA molecules that may be different than recipient HLA molecules. In lymph nodes, donor dendritic cells interact with recipient CD4 and CD8 T cells. Recipient T cells recognize donor HLA molecules or peptides derived from donor HLA molecules as foreign antigens. This can result in activation of adaptive immune response. Immune response can be cytotoxic or cell-mediated in nature that results in activation of cytotoxic cells like macrophages, natural killer cells, and CD8 T cells. Our immune response can be antibody mediated response. In this case, antibodies against donor HLA molecules bind to donor HLA molecules. This results in activation of complement and complement dependent damage to donor cells. Most of damage caused by antibodies to donor cells is mediated by complement dependent cytotoxicity. HLA antibodies that result in transplant rejection can be present even before transplant. These preformed antibodies can result in hyperacute rejection or acute rejection. And people who have preformed HLA antibodies also have higher incidence of chronic rejection and higher incidence of graft loss. Similarly, these antibodies can also be synthesized after transplant. And antibodies that are synthesized after transplant can result in acute rejection and chronic rejection. Most of antibodies that result in transplant rejection are directed against donor HLA class 1 and class 2 antigens. So most common target antigens that result in transplant rejection are donor HLA class 1 and class 2 antigens. However, other antigens can also contribute in transplant rejection. These antigens include minor HLA antigens and non-HLA antigens. Non-HLA antigens include angiotensin receptors and ABO blood group antigens. In next step, recipient T cells recognize donor HLA molecules or peptides derived from donor HLA molecules. This process is called allorecognition. Dendritic cells have HLA class 2 molecules and HLA class 1 molecules on their cell surface. These cells present antigens derived from pathogens on HLA class 2 molecules to CD4 T cells. T cells are capable of recognizing self-HLA class 1 and class 2 molecules, and immune response is directed only against antigens derived from pathogens. However, donor dendritic cells have donor type of HLA molecules and accompanying peptide. When these cells interact with recipient T cells instead of peptide recognize different epitopes of donor HLA molecule as foreign antigens. This is called direct allorecognition. In this type of allorecognition, intact donor HLA molecule present on a donor cell is recognized by recipient T cell. This type of allorecognition is more common in early period after transplant and results in strong immune response. With time, donor dendritic cells are depleted in transplanted organ and are gradually replaced with recipient dendritic cells. Recipient dendritic cells can ingest cell parts of donor origin. These cell particles can have HLA molecules as well. These HLA molecules are broken down to smaller peptides. These peptides are then presented on recipient HLA molecules to recipient T cells. So, in indirect allorecognition, peptides derived from donor HLA class 1 and class 2 molecules are presented by recipient dendritic cells to recipient T cells. As cell particles are ingested from extracellular environment, so immune response can be like immune response for extracellular pathogens, resulting in production of antibodies. Both direct and indirect allorecognition can result in activation and proliferation of recipient T cells. Another type of allorecognition is by semi-direct pathway. In this type of allorecognition, donor HLA molecules get incorporated on surface of recipient dendritic cells. These intact donor HLA molecules are then recognized by recipient T cells. Let's see what happens when recipient CD4 T cells have recognized donor HLA molecules or peptides derived from donor HLA molecules. After this, there is interaction of co-stimulatory molecules and production of cytokines by these cells. This results in activation and proliferation of recipient CD4 T cells. These CD4 T cells or helper T cells then convert to subsets of helper T cells based on cytokines. Main response in presence of inflammation in early period after transplant is Th1 response. In this type of response, helper T cells convert to Th1 cells. These cells secrete cytokines that promote cell-mediated response, resulting in activation of cytotoxic cells like macrophages, CD8 T cells, and natural killer cells. Another type of response can be Th2 response. These cells promote antibody mediated response, resulting in production of antibodies against donor HLA antigens or other antigens that will result in antibody mediated rejection. Another type of response is Th17 response. This response usually accompanies Th1 response. These cells also promote immune response and also produce cytokines that recruit neutrophils and macrophages. So all these responses promote immune response that will result in transplant rejection. Another subset of helper T cells called regulatory T cells or Treg cells actually don't regulate immune response and promote tolerance. In early period after transplant, other types of responses are dominant, so we give induction immunosuppression to avoid acute rejection. However, with time, some degree of tolerance may develop in some cases. Now let's discuss about different types of transplant rejection, with transplant rejection in case of kidney transplant as a reference. Hyperacute rejection results from ABO incompatibility or due to presence of HLA antibodies in high titers. After revascularization during transplant procedure, these antibodies bind to antigens present on vascular endothelial cells of transplanted organ. This results in activation of complement and complement mediated damage to these cells, resulting in disruption of vascular endothelial cells. This leads to thrombosis of graft vessels and ultimately graft loss. This type of rejection can occur after kidney, heart, lungs, and occasionally after liver transplant. However, this type of rejection is rare as we perform cross-match before transplant to find if there are any donor-specific antibodies present. Acute rejection commonly occurs during early few months after transplant. Majority of cases of acute rejection are due to acute cellular rejection, also called T cell mediated rejection. Around 10 to 15% of cases of acute rejection are due to acute antibody mediated rejection. We give induction immunosuppression to prevent acute rejection. This induction immunosuppression also reduces incidence of acute antibody mediated rejection due to its effects on T cells. In past, acute rejection was very common, but nowadays its incidence has reduced significantly due to improvements in immunosuppression. In acute cellular rejection, recipient cytotoxic cells infiltrate parenchyma of transplanted organ. In case of kidney transplant, it results in interstitial inflammation and inflammation around renal tubules. Inflammation of vessels can also occur in more severe cases. In case of kidney transplant, histological findings of acute cellular rejection are interstitial inflammation and inflammation around renal tubules in grade 1, intimal arteritis in grade 2, and transmural arteritis in case of grade 3. If left untreated, acute cellular rejection can result in complete destruction of graft. Most of cases of acute antibody mediated rejection are due to antibodies that are directed against donor HLA class 1 and class 2 molecules. Typically, these antibodies bind to HLA class 1 and class 2 molecules present on vascular endothelial cells of capillaries. However, larger vessels can also be involved in more severe cases. This binding of antibodies result in activation of complement and complement mediated damage to vascular endothelial cells. Complement mediated inflammation also leads to recruitment of inflammatory cells including neutrophils. So acute antibody mediated rejection typically results in microvascular inflammation. However, larger vessels can also be involved in more severe cases. Characteristic histological features of acute antibody mediated rejection include dilatation of capillaries, swelling of endothelial cells, and margination of inflammatory cells including neutrophils. In case of kidney transplant, microvascular inflammation leads to inflammation of glomerular capillaries and peritubular capillaries. For diagnosis of acute antibody mediated rejection, we need to consider three criteria. We need to have histological evidence of acute tissue injury that will appear as microvascular inflammation or intimal or transmural arteritis in severe cases. Then we need to have evidence that this acute tissue injury is caused by antibody mediated process. This can be achieved by C4D staining. However, C4D staining may not be positive in all cases. Then we need to identify presence of donor specific antibodies in recipient. Similar features of acute antibody-mediated rejection are also present in case of lungs, liver, pancreas, intestine, and heart transplant. Like acute rejection, tissue injury is also present in case of chronic rejection. However, in case of chronic rejection, there is evidence of chronic tissue damage. Chronic tissue damage results in deposition of extracellular matrix, fibrosis, and proliferation of cells. In case of kidney transplant, chronic cellular rejection results in interstitial inflammation and fibrosis of parenchyma. In grade 2 rejection, fibrosis and inflammation is present in vessel wall, and there can be proliferation of cells in vascular intima. Chronic antibody-mediated rejection is one of the leading causes of graft loss in case of all solid organ transplant. Some of histological features of chronic antibody-mediated rejection are similar to acute antibody-mediated rejection that include microvascular inflammation and inflammation of larger vessels. However, in case of chronic antibody-mediated rejection, there is deposition of extracellular matrix, fibrosis, and proliferation of cells. Repeated injury and repair mechanism in vessel wall leads to condition called transplant vasculopathy. In this condition, there is proliferation of cells in vascular intima and fibrosis in vessel wall. This results in impairment of blood supply to parenchymal cells. There is also associated fibrosis in parenchyma of transplanted organ. This condition results in accelerated arteriosclerosis in case of heart transplant and condition called bronchiolitis obliterans in case of lungs transplant. In case of kidney transplant, deposition of extracellular matrix and fibrosis in basement membrane of glomerular capillaries result in condition called transplant glomerulopathy. Similar findings are also present in peritubular capillaries, resulting in multilayering of basement membrane of these capillaries. Then we need to have evidence that these changes are caused by antibody-mediated process, and we also need to identify donor-specific antibodies. In this section, we have discussed about role of innate and adaptive immune system in transplant rejection. First, there is activation of innate immune system. Then recipient T cells recognize donor HLA molecules and other antigens. Immune response can be cell-mediated in nature. In this type of immune response, recipient cytotoxic cells infiltrate parenchyma of transplanted organ. Or it can be antibody-mediated response. Antibodies bind to vascular endothelial cells, resulting in microvascular inflammation or inflammation of larger vessels. In early period after transplant, acute rejection is main risk of graft loss. To prevent acute rejection, we give induction immunosuppression and we assess recipient for presence of donor-specific antibodies to prevent antibody-mediated rejection. Chronic rejection is mainly due to chronic antibody-mediated rejection and is one of the main causes of graft loss in long term.